In episode 1, we loaded a molecule and measured it. But before a computer can filter, search, or model molecules at scale, it needs a way to represent them. Today we build the 3 representations that every later step depends on, and use one of them to measure how similar 2 molecules really are. There are 3 ways to see a molecule in code. As a graph, atoms connected by bonds, the raw structure. As a vector of descriptors, a list of numbers like weight, greasiness, and ring count, which is what machine learning models eat. And as a fingerprint, a long bit vector that flags which substructures are present, like a chemical barcode. Graph, numbers, bits. Let's build each one. First, the graph. Under the hood, RDKit stores a molecule as atoms and bonds, nodes and edges. We can walk them directly. Aspirin has 13 atoms and 13 bonds. And if we draw it with the atom indices switched on, you can see the graph itself, every atom numbered, every bond a connection. This raw structure is the ground truth everything else is computed from. Second, descriptors. A descriptor is just a number computed from the structure, and if you stack several together, a molecule becomes a vector. Here's aspirin as several numbers: molecular weight, log P, polar surface area, rotatable bonds, aromatic rings, and hydrogen bond counts. That vector is exactly what a machine learning model needs as input, which is where we're headed in episode 6. Third, and most important for searching, the fingerprint. A Morgan fingerprint scans the neighborhood around every atom and flips on a bit for each substructure it finds, producing a fixed-length bit vector, here, 2048 bits, of which 24 are on for aspirin. Each lit cell means a chemical pattern is present. It's a molecule's barcode, and barcodes are easy to compare. Which brings us to similarity. Take 2 fingerprints and compute the Tanimoto coefficient. 0 means nothing in common, 1 means identical. Aspirin versus salicylic acid, its close chemical cousin, scores 0.45. Aspirin versus caffeine, a totally different molecule, scores just 0.09. That single number is the entire engine behind similarity search, finding new molecules that look like ones you already know work. So a molecule is a graph, a vector of numbers, and a fingerprint, and Tanimoto turns, are these alike, into a number. These representations power everything ahead: filtering, similarity search, and machine learning. Next episode, we stop using toy molecules and pull real data, hundreds of known EGFR drugs, from ChEMBL. See you there.